Molecular transformation involves the movement of discrete amounts of DNA into cells, the expression of genes on the transported DNA, and the sustainable replication of the transforming DNA. Plasmid or vector transformation, the process by which exogenous DNA is transferred into the host cell. Transformation usually implies uptake of DNA into bacterial, yeast, or plant cells, while transfection is the term usually reserved for mammalian cells. Typically, the method for transformation of a DNA construct into a host cell is chemical transformation, electroporation, or particle bombardment. In chemical transformation, cells are made competent, able to take up exogenous DNA by treatment with divalent cations, such as calcium chloride, which make the bacterial cell wall more permeable to DNA. Heat shock is used to temporarily form pores in the cell membrane, allowing transfer of the exogenous DNA into the cell. In electroporation, a short electrical pulse is used to make the bacterial cell temporarily permeable. Particle bombardment is typically used for the transformation of plant cells. Gold or tungsten particles are coated with the DNA construct and physically forced into the cell by gene gun. Traditional cloning, also called PCR cloning, required the use of the polymerase chain reaction, PCR, to get the multiplied number of copies, amplify the template sequence of the interest, usually the gene of interest, and add restriction sites to the ends of the sequence. Restriction enzymes are used to cut both the template of interest and the target vector, whereas DNA ligase is used to join the sticky ends of the template and vector together. Traditional cloning allows for flexible DNA sequence manipulation, which facilitates the building of nearly any desired construct. However, the checkpoints and optimization procedures required for traditional cloning can be cumbersome, and the reagents required can be expensive. TA cloning, called so due to the ability of adenine A and thymine T complementary base pairs on different DNA fragments to hybridize, and in the presence of ligase become ligated together, is one of the simplest forms of cloning. In this method, vectors containing 5' thymine overhangs are used to accept PCR products in which additional 3' adenosine overhangs have been added on by the nature of Taq polymerase amplification. TA cloning has the advantage of ease and speed since no restriction digestion step is required. In addition, TA cloning kits contain reaction buffer that contain the premix vector, ligase, and buffer, cutting ligation reaction time to as few as 5 minutes. The disadvantage of TA cloning technology is that the cloning is not directional, meaning the gene of interest may be inserted into the target vector in either the sense or antisense orientation. Normally, half of the subsequent transformants will contain the gene in the sense direction and half will contain the gene in the antisense direction. However, cells transformed with toxic genes may all display the genes in the antisense direction, since cells containing the sense-directed genes will not survive. In addition, survivor cells containing toxic genes oriented in the sense direction may be mutated to encode a less toxic protein. Seamless cloning technologies eliminate the requirement for restriction enzymes. This can be advantageous when an insert contains a number of restriction sites within its sequence, making it difficult to identify restriction enzymes that will not cut the gene of interest during the cloning procedure. Seamless cloning takes advantage of homologous recombination. There are numerous variations on the technique. In general, the procedure consists of adding flanking sequences approximately 15 base pairs in length to both the insert and vector via PCR. Exonucleases are used to chew back the insert and vector sequences, and the DNA joins use the recombinase enzymes or DNA ligase. Seamless cloning has been simplified by the development of kits that already contain a target vector and a proprietary mix of enzymes required for their recombinant reaction. For instance, seamless cloning GenScript's GenBuilder kit can clone inserts up to 10 kilobases in 30 minutes and can also be used for high-throughput cloning projects. Natural transformation is the bacterial adaptation for DNA transfer that depends on the expression of numerous bacterial genes whose products appear to be responsible for this process. In general, transformation is a complex, energy requirement developmental process. In order for a bacterium to bind, take up, and recombine exogenous DNA into its chromosomes, it must become competent, that is, enter a special physiological state. Competence development in Bacillus subtilis requires expression of about 40 genes. The DNA integrated into the host chromosome is usually, but with rare exceptions, derived from another bacterium of the same species and is thus homologous to the resident chromosome. In Bacillus subtilis, the length of the transferred DNA is greater than 1,271 kilobases, more than 1 million bases. The length transferred is likely double-stranded DNA and is often more than a third of the total chromosome length of 4,215 kilobases. It appears that about 7 to 9% of the recipient cells take up an entire chromosome. The capacity for natural transformation appears to occur in a number of prokaryotes, and thus far, 67 prokaryotic species in 7 different phyla are known to undergo this process. Competence for transformation is typically induced by high cell density and/or nutritional limitation conditions associated with the stationary phase of bacterial growth. Transformation in Haemophilus influenza occurs most efficiently at the end of the exponential growth as bacterial growth approaches stationary phase. Transformation in Streptococcus mutans, as well as in many other streptococci, occurs at high cell density and is associated with biofilm formation. Competence in Bacillus subtilis is induced toward the end of logarithmic growth, especially under conditions of amino acid limitation. Similarly, in Micrococcus luteus, a representative of the less well-studied Actinobacteria phylum, competence develops during the mid-late exponential growth phase and is also triggered by the amino acid starvation. Gibson assembly is a molecular cloning method which allows for the jointing of multiple up to 15 DNA fragments in a single isothermal reaction. It requires that the DNA fragments contain 20 to 40 base pair overlap with the adjacent DNA fragments. These DNA fragments are mixed with a cocktail of 3 enzymes: exonuclease, DNA polymerase, and DNA ligase, along with other buffer components. The resulting product is different DNA fragments joined into one. Isolinear or closed circular molecules can be assembled. There are 2 approaches to Gibson assembly: a one-step method and a 2-step method. Both methods can be performed in a single reaction vessel. The Gibson assembly one-step method allows for the assembly of up to 5 different fragments using a single-step isothermal process. In this method, fragments and the master mix of enzymes are combined, and the entire mixture is incubated at 50 centigrades for up to 1 hour. For the creation of more complex constructs with up to 15 fragments, or for constructs incorporating fragments from 100 base pairs to 10 kilobases, the Gibson assembly 2-step approach is used. The 2-step reaction requires 2 separate additions of master mix, one for the exonuclease and annealing step, and second for the DNA polymerase and ligation steps. For the 2-step approach, different incubation temperatures are used to carry out the assembly process. Functional cloning. is a molecular cloning technique that relies on prior knowledge of the encoded protein sequence or function for gene identification. In this assay, a genomic or cDNA library is screened to identify the genetic sequence of a protein of interest. Expression cDNA libraries may be screened with antibodies specific for the protein of interest or may rely on selection with the protein function. Historically, the amino acid sequence of a protein was used to prepare to generate oligonucleotides, which were then probed against the library to identify the gene encoding the protein of interest. Once candidate clones carrying the gene of interest are identified, they are sequenced and their identities confirmed. This method of cloning allows researchers to screen entire genomes without prior knowledge of the location of the gene or the genetic sequence. This technique can be used to identify genes that encode similar proteins from one organism to another. Similarly, this technique can be paired with metagenomic libraries to identify novel genes and proteins that perform similar functions, such as the identification of the novel antibiotics by screening for beta-lactamase activity or selecting for growth in the presence of penicillin. Please feel free to leave your questions and comments in the forum, and we'll be happy to answer them. Good luck, and see you soon. AJ.